Chapter 17, Breathing and Exchange of Gases. As you have read earlier, oxygen is utilized by the organism to indirectly break down nutrient molecules like glucose and to derive energy for performing various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reactions. It is therefore evident that oxygen has to be continuously provided to the cells and CO2 produced by the cells have to be released out. This process of exchange of oxygen from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following section of this chapter. Respiratory organs. Mechanism of breathing vary among different groups of animals depending mainly on their habitats and level of organization. Lower invertebrates like sponges, coelentrates, flatworms, etc. exchange oxygen with CO2 by a simple diffusion over their entire body surface. Earthworms use their moist cuticle and insects have a network of tubes, tracheal tubes to transport atmospheric air within the body. Special vascularized structures called gills are used by most of the aquatic arthropods and mollusks, whereas vascularized bags called lungs are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin also. Mammals have a well developed respiratory system. Human respiratory system. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is the common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is the cartilaginous box which helps in sound production and hence called the sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity which divides at the level of fifth thoracic vertebra into a right and left primary bronchi. Each bronchi undergo repeated divisions to form the secondary and tertiary bronchi and bronchioles, ending up in very thin terminal bronchioles. The tracheae, primary, secondary, and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular walled and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles, and alveoli comprises the lungs. We have two lungs which are covered by a double layered pleura with pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is in close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The parts starting with the external nostrils up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their ducts form the respiratory or exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clears it from foreign particles, humidifies and also brings the air to body temperature. Exchange part is a site of actual diffusion of oxygen and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber and which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by a vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side of the dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung cavity. Such an arrangement is essential for breathing as we cannot directly alter the pulmonary volume. Respiration involves the following steps: breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2 rich alveolar air is released out. Diffusion of gases, oxygen and CO2 across alveolar membrane, transport of gases by the blood, diffusion of oxygen and CO2 between blood and tissues, utilization of oxygen by the cells for catabolic reactions and resultant release of CO2. Mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs intrapulmonary pressure is less than the atmospheric pressure, i.e., there is a negative pressure in the lungs with respect to the atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal, intercostal between the ribs, help in generation of such gradient. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the anterior-posterior axis. The contraction of external intercostal muscles lifts up the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. That is inspiration. Relaxation of the diaphragm and the intercostal muscles return the diaphragm and sternum to their normal position and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, that is expiration. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times per minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. Respiratory volumes and capacities. Tidal volume. Volume of air inspired or expired during a normal respiration. It is approx 500 milliliter. That is, a healthy man can inspire or expire approximately 6,000 to 8,000 milliliter of air per minute. Inspiratory reserve volume. Additional volume of air a person can inspire by a forcible inspiration. This averages 2500 milliliter to 3000 milliliter. Expiratory reserve volume. Additional volume of air a person can expire by a forcible expiration. This averages 1000 to 1100 milliliter. Residual volume. Volume of air remaining in the lungs even after a forcible expiration. This averages 1100 milliliter to 1200 milliliter. By adding up a few inspiratory volumes described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity. Total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume. Expiratory capacity. Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume. Functional residual capacity. Volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity. The maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV and IRV, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity. Total volume of air accommodated in the lungs at the end of a forced inspiration. This includes RV, ERV, TV and IRV, or vital capacity plus residual volume. Exchange of gases. Alveoli are the primary site of exchange of gases. Exchange of gases also occur between blood and tissues. Oxygen and CO2 are exchanged in these sites by simple diffusion, mainly based on pressure or the concentration gradient. Solubility of the gases as well as the thickness of the membrane involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is represented as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressure of these two gases in the atmospheric air and the two sides of diffusion are given in table 17.1 and in figure 17.3. The data given in the table clearly indicates a concentration gradient for oxygen from alveoli to blood and blood to tissue similarly. A gradient is present for CO2 in the opposite direction, that is from tissues to blood and blood to alveoli. As the solubility of CO2 is 20 to 25 times higher than that of oxygen, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher compared to that of oxygen. The diffusion membrane is made up of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in our body are favorable for diffusion of oxygen from alveoli to tissues and that of CO2 from tissues to alveoli. Transport of gases. Blood is the medium of transport for oxygen and CO2. About 97% of oxygen is transported by RBCs in the blood. The remaining 3% of oxygen is carried out in a dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, whereas 70% of it is carried as bicarbonate. About 7% of CO2 is carried in a dissolved state through plasma. Transport of oxygen. Hemoglobin is a red colored iron containing pigment present in the RBCs. Oxygen can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of oxygen. Binding of oxygen with hemoglobin is primarily related to partial pressure of oxygen. Partial pressure of CO2, hydrogen ion concentration, and temperature are the other factors which can interfere with this binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with oxygen is plotted against the PO2. This curve is called the oxygen dissociation curve and is highly useful in studying the effect of factors like PCO2, H plus concentration, etc. on binding of oxygen with hemoglobin. In the alveoli where there is high PO2, low PCO2, lesser H plus concentration and lower temperature, the factors are all favorable for the formation of oxyhemoglobin, whereas in the tissues where low PO2, high PCO2, high H plus concentration and higher temperature exist, the conditions are favorable for dissociation of oxygen from oxyhemoglobin. This clearly indicates that oxygen gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 milliliter of oxygenated blood can deliver around 5 milliliter of oxygen to the tissues under normal physiological conditions. Transport of carbon dioxide. CO2 is carried by hemoglobin as carb-aminohemoglobin about 20 to 25%. This binding is related to the partial pressure of CO2. PO2 is a major factor which could affect this binding. Even when PCO2 is high and PO2 is low, as in the tissues, more binding of carbon dioxide occurs, whereas when the PCO2 is low and PO2 is high, as in the alveoli, dissociation of CO2 from carbaminohemoglobin takes place. That is, CO2 which is bound to hemoglobin from the tissues is delivered at the alveoli. RBCs contain a very high concentration of the enzyme carbonic anhydrase and minute quantities of the same is present in the plasma too. This enzyme facilitates the following reaction in both directions. At the tissue side where partial pressure of CO2 is high due to catabolism, CO2 diffusion into blood, RBCs and plasma and forms bicarbonate and H plus at the alveolar site where PCO2 is low and the reaction proceeds in the opposite direction, leading to the formation of CO2 and water. Thus, CO2 trapped as bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Every 100 milliliter of deoxygenated blood delivers approximately 4 milliliter of CO2 to the alveoli. Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system, a specialized center present in the medulla region of the brain called respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the functions of the respiratory rhythm center. Neuron signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process by which these substances can be eliminated. Receptors associated with aortic arc and carotid artery also can recognize changes in CO2 and H plus concentration and send necessary signals to the rhythm center for remedial actions. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. Disorders or respiratory disorders of respiratory system. Asthma is a difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased. One of the major causes of this is cigarette smoking. Occupational respiratory disorders in certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation leading to fibrosis, proliferation of fibrous tissues, and thus causing serious lung damage. Workers in such industries should wear protective masks. Thanks for watching.